Thank you very much for that introduction. Thank you for the invitation. Really happy to be here in London today. I'm not a physiologist, as you heard, but I guess everything is physiology, so maybe, hopefully, something that I have to say today will resonate with some of you. So I'm going to talk to you today about your most important tissue. Those of you who know me know what that tissue must be. That would be muscle. I often hear from my neuroscientist friends how important the brain and the nervous system is, but really what I tell them is you can live without a brain. Teenagers do it all the time, but there is no life without muscle. Muscle is responsible for the wonders of humanity, from the beating of the heart once per second from embryogenesis to adulthood, which defines the boundaries of life itself, to many of the wonders of biology, the beating of the hummingbird wing 95 times per second, to the movement of the largest animals that ever roamed the earth, all aspects of art, music. There's my friend Willie Nelson up there in the right-hand corner. To athleticism, there's Roger Federer at Wimbledon, where I was fortunate to attend last year. Every aspect of life really depends on the functioning of muscle. And so today I'm going to talk to you about just one muscle in particular. I'm talking to you about skeletal muscle, which is the largest tissue in the body. It accounts for 40% of human body mass. It's, as I mentioned, it's the sole output of the nervous system. The nervous system can do all at once, but without muscle, there is no readout. And it's the most regenerative of all tissues. And over the past 3 decades, the students and postdocs in my laboratory have discovered many of the key regulatory genes, transcription factors, and molecules responsible for the formation of all of the muscles of the body, including skeletal muscle. So over the past decades, we've focused on the discovery of new proteins. We've discovered many microRNAs that modulate facets of muscle biology and disease. And in recent years, we and others have turned our attention to a large and poorly understood class of RNAs called long non-coding RNAs, and we're continuing to work on those. But what I want to talk about today is one facet of non-coding RNAs, and that is that many of them unexpectedly actually are not non-coding at all, but they encode tiny peptides that play fundamental roles in physiology and biology and development. And this work that I'll talk to you about in the first half of my talk was carried out by 2 postdocs, Kat Makarowicz and Doug Anderson, and one graduate student, Ben Nelson, as they mined the microproteome. This is one of my favorite prints from René Magritte, which hangs on the wall of our laboratory. And I think this was really an influence on the work that I'm going to talk to you about today. In this painting, which will be familiar to many of you, the artist looks at the egg, and he sees beyond the egg to what could be, and that is the bird. And I'm continually telling my students and postdocs not to work on what everybody else is working on. Don't look at what everyone else is doing. Look beyond what everyone else is doing. And so I think this painting, which sits above the microscope in our lab, really has been influential, at least in my thinking. So we began to try to look at non-coding RNAs with unexplored roles in muscle biology, and this was one of the ones that we began working on. This is an RNA blot. You can see why we were interested in this putative non-coding RNA. It's exquisitely specific for skeletal muscle and not expressed anywhere else. But the more we worked on this, the less we could figure out about its function. And so Doug Anderson, the postdoc I showed you previously, really began to try to look beyond the superficial, and he recognized that there was a short open reading frame, unannotated and coded by this non-coding RNA, and it was only then that we appreciated the fact that open reading frames less than 100 amino acids are not annotated in databases. And that's because it's too difficult to discern real peptide sequences from noise when you get down to 100 amino acids or less. And so we named the micropeptide encoded by this RNA as myoregulin for reasons that you'll see. Now, myoregulin here is an abbreviated MLN. It's a tiny peptide, about 44 amino acids long. It had no homology in the database, but we recognized that structurally it was likely to have a short transmembrane region and a cytoplasmic domain, and we looked for other proteins with similar structures, and there were 3 of those that had been previously characterized biochemically. One is a famous protein called phospholamban, abbreviated here as PLN, a related protein called sarcolipin, SLN, and the ancestral member of this family, sarcolamban, in insects. And there was no overt homology between these proteins, except we noted that there were 3 residues here, LF, XXF, in the putative transmembrane region that were identical across these 3 proteins. And so we were curious as to whether myoregulin might have a function similar to these other proteins. And these other proteins, many of the physiologists in the room will know, have been shown to function as regulators of the main calcium pump, the sarcoplasmic calcium ATPase, SERCA. So muscle contractility, which I do not need to educate the physiologists in the audience, of course, is controlled by the cycling of calcium from the lumen of the endoplasmic reticulum, sarcoplasmic reticulum in muscle, to the cytoplasm. Calcium exits through the ryanodine receptor, binds to the sarcomere, induces contraction, and then is pumped back into the SR by the SERCA, the calcium pump. And this is the molecular basis, of course, for contractility. And the 2 former members of this family that had been identified, phospholamban and sarcolipin, shown here, have been shown to interact with the SERCA pump, and they function as brakes on the calcium pump. And this gives the SERCA pump a greater dynamic range of activity by having this inhibitor present. So we went back and we wondered whether these 3 residues, LF, XXF, might have any role in the interaction between these proteins in SERCA. Remarkably, those residues had never been studied despite decades of work on phospholamban. So this is an immunoprecipitation assay. It's a little complex, but suffice it to say that if we immunoprecipitate myoregulin, MLN, with SERCA, it will bring down SERCA in a complex. But if we mutate any of these 3 residues, L, F, F, to alanines, completely abrogates the interaction. So those 3 residues constitute at least part of the molecular determinants for the physical interaction between this micropeptide and this calcium pump. And we did molecular modeling of myoregulin and SERCA, and that's shown here. So this is the model of SERCA in gray, and this is where sarcolipin is known to interact with a tiny groove in the transmembrane region. And myoregulin occupies exactly the same groove, suggesting that it might fulfill a similar function. Now this was really interesting because it began to at least hint at an explanation for a long-standing paradox in the muscle field, and that is that SERCA controls contractility of every muscle in the body, but its 2 negative regulators, sarcolipin and phospholamban, shown here by expression analysis, are only expressed in the heart, essentially. And this had raised the question, are there regulators of the SERCA pump in skeletal muscle, or does the SERCA pump function completely autonomous of this type of inhibitory mechanism? And it turns out that myoregulin was expressed specifically in all muscle types in the body and nowhere else. And so we believed that this myoregulin peptide could fulfill the similar functions of these other SERCA regulators in skeletal muscle. So to look at that, we generated knockout mice lacking this micropeptide. And to do that, we used a strategy where we introduced a tomato reporter. This is to mark the expression pattern of the gene, and we put after it 3 poly-A sequences which terminate transcription of the gene. And so the third exon of this gene, shown in red, encodes the tiny micropeptide. So if you insert this stop cassette in exon 1, it eliminates production of the protein. And so then we looked at these mice, and they were homozygous viable, and this really frustrated us, frankly, because here we discovered this, what we thought was a really interesting protein, and we knocked it out and no function. And these mice were viable, and we thought, okay, if calcium cycling is important for muscle function, maybe they have some kind of function in regulating muscle contractility. And it turns out, here you can see that the top is a wild-type mouse, and the bottom is a myoregulin knockout subjected to wheel running. This is forced treadmill running. And you can see that the wild-type is starting to falter here, and the knockout just keeps going. And so elimination of this negative regulator, this brake on the circuit pump, confers an endurance phenotype on these animals and essentially makes them marathon runners. So that was the loss of function consequence. We also did the gain of function consequence. We overexpressed myoregulin in muscle, and it causes a severe myopathy. So you can see that here. Myopathy is often characterized by central nuclei and heterogeneity of muscle fiber diameters, and you can see that in these mice. So we think that muscle fibers maintain this tiny micropeptide at very precise levels to modulate muscle contractility and function. So coming back then to the model of muscle contractility, here's the circuit pump, the central regulator of calcium homeostasis of muscle. We believe its activity is negatively regulated by its physical interaction with this tiny micropeptide in the transmembrane region, and that in this way, myoregulin fulfills a function similar to that of phospholamban and sarcolipin. So this was interesting. But there was another paradox, and the physiologists in the audience will know this better than me. That is that circa, the calcium pump, exists in every cell in your body, not just in muscle. It's a major regulator of calcium homeostasis by controlling cycling of calcium from the cytoplasm to the nucleus. And so I told you that there are two cardiac members of this family of circa regulators, sarcolipin and phospholamban. And I told you that there's another micropeptide regulator, myoregulin, 44 amino acids in length. But we wondered, could there be other ones that have gone undetected all these years? And so we actually went back to a database that we had generated of non-coding RNAs, and we interrogated that for any potential codons that could encode an LFXXF sequence of amino acids. And lo and behold, we found another one. And this was a non-coding RNA, misannotated as such, and here's its sequence. We called this peptide endoregulin because it turned out it's expressed specifically in endothelial cells, and it has the same LXXF motif. And so we wanted to keep going. And we went back, we found another one. And we were running out of names at this point, so we called this another regulin. And you can see it's also a tiny peptide, also misannotated as a non-coding RNA, and this one is broadly expressed in all tissues in which the other four members of the family are not expressed. So I don't want to get into too many of the details about these peptides, which we call regulins, but there's something really fascinating about them, and that is that there are multiple isoforms of the circa calcium pump. There's 5 of them to be precise, and there's 5 regulins that I just described to you. And each circa isoform has evolved to be expressed in one particular tissue with its regulin micropeptide. So you can see that the detail is complex and the details aren't that important, but here you can see this is expression data showing the tissue distribution of circa 2A and phospholamban and sarcolipin. These are the 2 cardiac-specific micropeptides that interact with circa 2A, which you can see here is also largely cardiac-specific. And this shows the expression pattern of myoregulin, which you already know from what I showed you is skeletal muscle specific. It turns out there's one circa isoform co-expressed with it, circa 1. And here's endoregulin, the endothelial restricted regulin, shown here in these peaks. It's confined to these tissues, and there's one circa isoform, circa 3, that's expressed with it in that tissue, those tissues. And then the last one's the ubiquitous one, ALN, another regulin. It's co-expressed with SERCA2B. So I think this is really interesting, and it just makes you wonder whether there was an ancestral partnership of one SERCA and one regulin in an ancient species. And as those two genes duplicated, the regulatory elements that controlled them duplicated as well to confer the unique expression of each of these in different tissues. And so this is a summary of those different genes and their expression patterns and their partnerships. And so we're currently using knockout mice gain-of-function studies in biochemistry to further explore how these different regulin micropeptides modulate perhaps not only calcium homeostasis, but other properties of the cells in which they're expressed. So I want to mention one more micropeptide before I move on, and that is a protein, a micropeptide shown here, which we named Dwarf for Dwarf Open Reading Frame. This was also annotated as a non-coding RNA, and you can see why we were interested in it. It's only expressed in the heart, and it turned out that this was not a non-coding RNA at all. It encodes a micropeptide. It's the third smallest known protein encoded by the human genome. It's 34 amino acids in length. Now, for the physiologists in the audience, I should clarify a point, and that is all of us are used to thinking about tiny peptides. Many hormones are processed from large precursors to give rise to small, tiny peptides. The types of micropeptides I'm talking to you about today are not like that. These are micropeptides that are encoded by a standalone open reading frame from the methionine to the stop codon with no processing. They just generate these tiny peptides, so they're unlike large pre-protein precursors. And so this micropeptide has also a transmembrane region. You can see here, and it barely has anything beyond the transmembrane region, just about 9 or 10 residues or so. We raised an antibody against Dwarf, against that tiny N-terminal peptide, and we stained heart cells with it, and here you can see the expression pattern. And lo and behold, the Dwarf protein superimposes in its expression pattern over SERCA. Again, a membrane-bound protein that looks like it interacts with SERCA. And if you just express the two proteins together, this is in COS cells, misexpressing them, they co-express perfectly in membranes. So we speculated that Dwarf might also interact with SERCA, and since this work's published, I'm just going to summarize the conclusions of this work. That is that Dwarf turns out to be yet another partner protein for the SERCA calcium pump. But in contrast to all the ones I've described to you previously, Dwarf functions as an activator of the SERCA pump. It stimulates calcium cycling through this calcium ATPase, and it does so, we believe, at least in part, by occupying the same transmembrane region that the negative inhibitory regulins typically occupy. So at least part of its mechanism of action is through displacement of the inhibitory peptides, but it might have other roles as well. Now, calcium cycling and the SERCA pump are known to be fundamental regulators of cardiac contractility that are dysregulated in heart failure. And there's been a lot of efforts by pharmaceutical companies to develop activators of the SERCA pump to enhance cardiac contractility in the setting of heart failure. So we decided to look at Dwarf expression and function in the setting of heart failure, and this was quite interesting. This is Western blot with an antibody to Dwarf. You can see it's about 7 kilodalton micropeptide that's in 6 different mouse hearts. And several years ago, Jeff Mulcantine, when he was a postdoc in our lab, made mice that overexpress calcineurin, calcium phosphatase, in the heart, and they have really profound heart failure. And you can see that in that heart failure model of the mouse, there's no expression of Dwarf. Dwarf is completely silenced in its expression. So then we looked in human failing hearts, and also Dwarf expression was dramatically downregulated in the failing human heart. So we wondered, okay, this is an activator of the SERCA pump, which would be predicted to augment cardiac contractility, and it's vanishing in failing hearts. Might there be a functional connection? So we decided to look at that. Now, there are many models of heart failure. Perhaps the most dramatic, one of the most dramatic models is a model of mice that lack a muscle limb domain protein, abbreviated MLP. And these mice have been shown by others to have severe dysregulation of calcium cycling. And as a consequence of the perturbation in calcium handling, these hearts become massively dilated, and you can see the sarcomeres are extremely disrupted. And so we just took a long shot guess and asked, would it be possible to rescue this most dramatic mouse model of heart failure due to calcium abnormalities by forcing the expression of MLP to normalize calcium homeostasis through the pump. So these are echo tracings in wild type. Each shows the contractility of the heart. The MLP mice, it's amazing these mice are even alive. When you look at their echoes, they're basically flatline. They have almost no cardiac contractility. So what we did was we made a mouse where we overexpressed Dwarf under a separate promoter that would be insensitive to the endogenous signaling, and we overexpressed it in the heart, and we crossed it into the MLP knockout. Here's what we found. The simple forced expression of this micropeptide completely restored cardiac contractility in this model of genetic heart failure. And you can measure that by ejection fraction, a measure of cardiac contractility. So in the MLP knockout mice, ejection fraction is down about 20%, and if you force the expression of Dwarf in these hearts, it brings the ejection fraction back to normal. And here you can see the histology. So the middle, these animals are in failure. Their hearts are dilated. If you force the expression of Dwarf in the heart, it restores normal cardiac architecture. And here you can see fibrosis. In MLP knockout mice, there's extensive fibrosis. That's shown here by staining with picocereus red. And by expressing this one micropeptide, you can restore architecture and block fibrosis. So we believe that this cardiac-specific micropeptide Dwarf functions at a nodal point in the pathway for calcium regulation in the heart, and that by elevating Dwarf expression or augmenting SERCA activity, one can stimulate calcium cycling and bypass many of the structural and functional abnormalities that are associated with genetic deletion of this one particular sarcomere protein, MLP. We're currently looking at other genetic and various models of heart failure for their sensitivity to SERCA activation by Dwarf. So what I've told you so far is that there is a multitude of unrecognized micropeptides out there that have not been annotated. I've told you about a few that function in sarcoplasmic reticulum to modulate the SERCA pump, but I'll tell you that there are many others that we are working on right now, some of which are secreted, and we believe they encode proteins involved in intertissue communication. Others we think function as cofactors. Some are in the nucleus, and we don't really know what all the functions of these are. So, as you could probably figure out by now, I think about biology as art. And so in thinking about this is a famous print. This is on the wall of my office, actually. This is sort of how I think about micropeptides in biology. I mean, we're used to thinking about multi-domain, complex proteins, enzymes, channels, scaffolding proteins, transcription factors. Micropeptides are singular tiny protein domains. That's essentially what they are, one tiny domain. And what they do is they can insert themselves in various ways into nooks and crannies of larger proteins, and they can thereby modulate their functions. And so we are currently exploring this, and we think it represents an exciting new frontier for identifying unrecognized cofactors, coactivators, allosteric modulators, ligands, etc., for more complex proteins encoded by the genome. So I want to shift gears now to the second half of my talk and tell you about diseases of muscle. So I think it's humbling. It's also an opportunity to realize that muscle is subject to a multitude of monogenic disorders. Many of these are the most devastating disorders of mankind. There's more than 840 monogenic neuromuscular disorders in more than 450 genes with all sorts of functions, everything from mitochondrial genes to enzymes to scaffolding proteins, and you name it. There's mutations that cause muscle disease. And so we have decided to try to address this major unmet clinical need. And the work I'm going to talk to you about today was carried out by 4 individuals in my lab, Chengzu Long and Leonella Amoasi. Chengzu is a student, Leonella is a French postdoc, and Yu Zhang and Yi Li Min are 2 students. And we decided to tackle perhaps the most insidious of all muscle disorders that's familiar, I think, to all of you, and that is Duchenne muscular dystrophy, or DMD. This is a horrific disease, and the deeper I've gotten into this field and interacting with patients, the more I've become cognizant of that. The disease affects 1 in 5,000 boys. 3,000 boys worldwide suffer from this disorder. This is passed on from mother to son on the X chromosome, so the mothers are unknowing carriers. It's typically diagnosed around the age of 2. Mothers will notice that their sons are having some difficulties walking. They go to the doctor and they ask to do the Gower's sign, which is they ask the boy to stand up, and they can't stand without the use of their arms because they have weakness in the leg muscles. And it rapidly progresses to loss of ambulation, confinement to wheelchair, ventilation, and ultimate death. The cause of death in these boys is cardiomyopathy due to degeneration of the heart. So the disease is caused by mutations in the largest gene in nature. This is the gene encoding the dystrophin protein. This is really massive. Dystrophin gene is on the X, as I mentioned. It spans 2.5 million bases. Just give you a sense of how big that is. Takes RNA polymerase 18 hours to traverse this gene one time. So it basically takes you all day, your entire day, you make 1 copy of this RNA. I mean, this is really a wonder of biological engineering. The gene encodes 79 exons, 3,500 amino acids, roughly. And here's one of the big challenges. There's more than 3,000 mutations that have been identified around the world in boys with this disorder. And I get every day multiple emails from mothers every corner of this planet telling me what their son's mutation is. And almost all mothers, they find out what the mutation is, but there's nothing that can be done about it. So these are the way these mutations can be categorized. 2/3 roughly are exon deletions that put the protein out of reading frame. 1/3 roughly are point mutations that impose a stop codon or other deleterious amino acid in a functional domain, and then about 5% are duplications that disrupt the reading frame. So the gene encodes the dystrophin protein, which has been first discovered by Lou Kunkel, and there's been beautiful biochemistry done on this protein by Kevin Campbell and others. The dystrophin protein, you can think about it as a shock absorber for all muscle tissues, heart and skeletal muscle. It resides beneath the membrane, and it connects a multi-protein complex at the cell membrane called the dystroglycan complex with the cytoskeleton. And it's required to maintain the integrity of the muscle membrane during each cycle of contraction and relaxation. And in its absence, the muscle tears and leads to muscle degeneration. So here's what it looks like. This is an immunostain in green on the left, showing a muscle, skeletal muscle, and you can see it's homogeneously distributed. And you can think about it, again, I'll come back to this analogy throughout the talk, but it's like a shock absorber for muscle. So here's the exon structure, 79 exons, as I mentioned, and this will be really important for the non-experts in the audience for what I'll say for the rest of the talk. The shapes of these exons indicate whether the splicing between those exons can maintain the reading frame of the protein across that exon junction. So they have to be compatible to keep the reading frame. And you can see that the domains of the protein in the top are color-coded on the exons at the bottom. And another fascinating feature of this gene is that it's obviously ultra-complicated, and when nature invented it, nature didn't change it. So the structure of the gene and the intron-exon boundaries and splicing patterns and everything are all conserved from mouse to humans. So if you can figure something out in a mouse, you can extrapolate it pretty well to the human setting. And so coming back to the shock absorber analogy, if you think about a shock absorber, you have to have the two ends to anchor it. And the central region is a series of spring coils. The coils aren't all essential. Some of them, they're redundant, but you have to have the two ends to anchor the thing in place. And the reason we know that is because there's patients with a mild form of muscular dystrophy called Becker's muscular dystrophy. And these patients survive into their 70s and are ambulatory and they're really not so severely impaired. And what they have is in-frame deletions of some of the coils in the spring, but they still have some of the coils and they still have the anchoring regions. So over the past 25 years, there have been heroic efforts to try to find cures for this disorder, and I'll tell you, everything has failed. That's probably not a popular comment, but that's the fact. So here's some of the approaches that have been tried so far. Dystrophin-like gene delivery. There's some real promising results to deliver dystrophin mini genes with viruses. So far, that hasn't been tested in humans. It's a challenge because this gene is so big. To consolidate it into a virus is difficult. Exon skipping oligos have not worked. I'll have a comment about that in a moment. Upregulating of a compensatory gene called utrophin, in principle, could work. It hasn't yet been done in humans. There's been a lot of work done on myoblast and stem cell transfer. It's not going to work. 40% of your body mass is muscle. You cannot reconstitute the musculature with myoblasts. And one thing I think we've learned from the cardiac field over the past decade, that is, stem cells don't colonize the heart very well. And so to try to rescue cardiomyopathy in these boys with stem cells has not worked. And there's been other approaches that don't work. So I want to just highlight one approach. This has been really widely publicized in the States, maybe also in the UK. There's a drug called eteplirsen developed by Sarepta Therapeutics. Works in animals and in cells, but to some extent hasn't worked in people yet. This is an oligonucleotide. The way it works, shown right here, the most common deletion in boys is deletion of exon 50 of the gene. I'll have a lot to say about that later. But you can see when exon 50 is deleted in these boys, 49 splices to 51, and you see the shapes are incompatible, so it puts the protein out of frame, and then these boys can't make the other end of the shock absorber. So this drug is an oligo, and it masks the splice acceptor on exon 51. So in principle, that would allow 49 to splice to the next exon 52 and put the protein back in frame. This is called exon skipping. The drug did not show any efficacy in a clinical trial of 12 boys. It was an open trial with no controls. But there was nothing that could be done for these boys with the disease, so the FDA approved the drug, and it now costs roughly $1 million per year for boys to have this injected through an indwelling catheter into their hearts, and there's so far no evidence for efficacy. Hopefully that will change with time. But suffice it to say, there's been a need really for something that gets at the root cause of the disease, and that's the mutation. So we decided to try to apply gene editing to this approach, to this disease. And I think most or all of you know how CRISPR gene editing works. I won't go into all the details. It's a two-component system. It involves a bacterial enzyme, most commonly is called Cas9, but there's a whole host of these. And it involves a second component, a guide RNA, which can be used to deliver Cas9 to any region of the genome. And in principle, when Cas9 is delivered to a mutant region of the genome, it'll cut, and then the gene will get edited and can get fixed. So we decided to at least test in proof of concept studies whether this approach could be applied to Duchenne. And I don't want to get into all the nuts and bolts of this approach, but there's a couple of points I need to make about how it works. And so this is, I think all of you know, this is a bacterial immune system that the bacteria evolved to recognize invading viruses and to cut the DNA and eradicate them. So here's how it works. Cas9 will be directed to any sequence in the genome if you provide a short guide RNA, abbreviated here sgRNA, with complementarity to the sequence of interest. So when the guide RNA and Cas9 find their way to the target sequence, if there's one other component of the system, and that's a dinucleotide called the PAM sequence shown here, which is NAG or NGG. If those two sequences are adjacent to the region to which the guide RNA anneals, then Cas9 will cut. And it's incredibly efficient. Work from Jennifer Dowden and others have shown that Cas9 scans the genome and it finds its target and cuts within seconds and moves on to the next. It's really a remarkable mechanism. And if a DNA template is provided, then it will insert the DNA template and it'll correct the mutation by homologous recombination. If a DNA template's not provided, then it'll cut. And then the endogenous repair machinery of the cell will fix the cut, but it's not precise, so it leads to insertions or deletions, so-called indels at that region. So in the first proof of concept study, we used a mouse called the MDX mouse. This is the most widely used mouse model for muscular dystrophy. It harbors a single point mutation in the 23rd exon of the gene shown here that imposes a stop codon, so these mice can't make dystrophin. And so we initially, our thinking was not really so crystallized at that point. So we initially just asked if we could do germline editing to fix this mutation by homologous recombination. So in germline editing, what you do is you isolate fertilized oocytes from a mouse and put them in a dish, just sort of like exactly as you would do in an in vitro fertilization clinic. And then we injected these fertilized eggs with Cas9 and a guide RNA for this exon and a wild-type DNA sequence for that exon to ask if we could replace it. And then you put these embryos back in a mother, and she gives rise to birth. And when you do this, the offspring are mosaic. And that's because the editing takes place over time, and the early embryo is cleaving and giving multiple cells, and so they don't all end up being edited at the same moment. But it turns out that this mosaicism was really a bonus, because it enabled us to ask, what is the minimum level of editing that you need to correct this disease, at least in this mouse model? And so here's the answer. So these are muscles stained in green for dystrophin. Here's wild type. Here's the MDX mouse, has no dystrophin at all. Here's an animal. This animal only had 17% correction. At the gene level, we could sequence it and see what the percent correction was, but you could see it's pretty good. It has almost full restoration of dystrophin, not quite, but pretty good. And this animal has 41% gene correction. It's not real efficient, but they're completely normal. So the first take-home message from this study was that it's not necessary to correct every mutation in the body to fix this disease. Even a relatively small level of correction can be potentially effective. It's been calculated that in boys, if you could correct 15%, 15%, or restore 15% of dystrophin, that you could cure this disease. So it's not level to bring it back to 100%. Now we wondered why is the efficiency of restoration of dystrophin so much greater than the efficiency of gene correction? There's a disconnect, and I won't get into all the details, but the answer, I think, lies in the biology of muscle. That is, muscle is a syncytium, has thousands of nuclei in a common cytoplasm, and if you can correct even a few of those nuclei and induce them to produce dystrophin, that dystrophin can extend the range of correction. And also, muscle has two stem cell populations that I won't get into today, but we think if those stem cells get edited, they provide a permanent reservoir for the corrected allele. So after this first paper, there were a lot of issues raised, and appropriately so. One is that germline editing, as we had done in that paper, is not appropriate in people, although that may change at some point. But nevertheless, that's a really important point. Homology-directed repair, homologous recombination that we used in that first study does not occur in post-mitotic tissues. That's a biggie because muscle and heart don't divide, and so the machinery for homologous recombination doesn't exist in those cells. So you can't even use the method that we had used. Thousands of mutations. I told you already there's 3,000 mutations. How are you going to fix all these in a simple way? And then how are you going to deliver everything to all the muscles and the heart? So here's the structure of the gene again. Here's the major exon deletion in boys, deletion of exon 50. That's the deletion that the eteplirsen Sarepta drug was directed against. And when you can see when you delete that, you eliminate the C terminus, and so you get rid of that region of the protein. So there was no mouse to study this, so we made one. So in this case, we used CRISPR to delete exon 50 ourselves in mice. And just as in humans, here's the results. Dystrophin's in red here, so we call it the delta exon 50 mouse. Has no dystrophin at all. So it serves as a platform for testing any therapy, really, but we are focused on CRISPR. And one of the non-invasive diagnostic markers for muscular dystrophy is creatine kinase, CK, which circulates in the bloodstream when muscles are leaking it out. And you can see the deletion mouse has roaring levels of CK. So we looked at this and we thought, how can we fix this disorder? And then we had a kind of a eureka moment as we looked at the PAM sequence. You remember the PAM sequence is the dinucleotide that has to be adjacent to the guide RNA, it's AG or GG. And we looked at that and we realized that the AG is the universal splice acceptor sequence for all exons. Every exon in the world is preceded by an AG. Same exact sequence as Cas9 recognizes. So you might even start to think about intelligent design if you see this. So this bacterial gene editing enzyme uses the same sequence as the universal splice acceptor sequence, and so we thought we could just use this to do exon skipping by making an indel. So the thought was if you'd made a guide RNA and used this PAM sequence adjacent to any exon you want to skip, let Cas9 cut there and make an indel, and then let the cellular machinery fix it. And it won't fix it perfect. It'll give you indels at that region, but it doesn't matter. All you got to do is get rid of the splice acceptor for this mutant exon and let the cell take over and go to the next exon and put it back in frame. So that's what we tried to do, and we needed to get this into adult or newborn mice. We didn't want to do germline editing, so we engineered Cas9 and guide RNA into AAV, a harmless virus that has tropism for muscle and heart, and we injected this virus into these mice. And here's what we got. These are the first animals we looked at. So here's the tibialis anterior. It's a muscle of the leg. In the middle is the mouse that has the deletions. You can see the muscles basically falling apart. These mice are about 6 weeks old, I think. And then on the far right are animals 4 weeks after we injected the gene editing components, and muscle histology was restored to normal. This is immunostaining for dystrophin, and these are two representative muscles. Every muscle looks the same. Here's the mouse model lacking exon 50, so dystrophin production is abrogated. And then there's 4 weeks after injection, dystrophin is restored. I told you that dystrophin interacts with a multi-protein complex at the membrane called the dystroglycan complex. Here's two of the components. And in the absence of dystrophin, those components don't accumulate at the membrane, but if you do exon skipping to restore dystrophin, now you can restore the dystroglycan complex in these muscles. And here's the heart. So left panel is dystrophin and all the cardiomyocytes. The middle panel is the humanized human model for exon 50 deletion, no dystrophin, and then 4 weeks after one systemic dose of CRISPR-Cas, dystrophin is restored. And this is quantification. I think it's really important in these approaches to quantify the amount of dystrophin that is produced. In the Sarepta eteplirsen trial, they were asked to do that in these boys, and they were not able to demonstrate dystrophin protein above 1%, plus or minus 1%. So really there was no statistical evidence for dystrophin protein. This is Western blot in duplicate in the hearts. So normal dystrophin, here's the mouse, no dystrophin, and here's 4 weeks after injection with gene editing components. About 80% of the level of dystrophin protein is restored. And then here's muscle strength. So you can measure muscle strength in a number of ways. In this case, we did it by measuring grams of force. The mice with the exon 50 deletion are impaired, and here, 4 weeks after injection, the muscle strength, it's not back to normal, but it's moving in the right direction. So this is the structure of the gene again. The exons in red indicate hotspots for mutations in humans. So about 60 to 80% of the human mutations basically, well, it's about 60% actually, localized to these top 10 exons in red. So if you could skip these exons, you could correct a very large percentage of boys with Duchenne. And so we screened for and identified guide RNAs, the optimum guide RNAs from the human genome, to direct Cas9 to each of these exon splice acceptor sequences to induce skipping to reframe the open reading frame in boys with these mutations. So here's what we're trying to do. Patients come in, and to Dallas, we have a muscular dystrophy center. We can take a small sample of their blood, and we can make IPS, induced pluripotent stem cells, from the patient. We can then use these guide RNAs to ask, can we correct the mutation in cells from these boys? And then we can convert them to muscle in a dish. It's a real simple method, and look at dystrophin protein. So initially, we were taking blood samples from boys and just testing this approach on various mutations. But there's so many patients, and there's so many, a lot of them are just in distant corners of the earth where we can't get. So if a mother writes and says, my son has this mutation and it's an interesting one, or we don't know if we can fix it, or we haven't done it, we'll just make the mutation in human iPS cells, and then we can test it by this approach. So you don't really have to go to the patient. So here's just an example. So this is a real boy. This boy's missing exon 50, so again, we deleted the exon 51 splice site. Here's his cardiomyocytes in red, stained for dystrophin. There's no dystrophin in green. And here, by a single step of skipping, we can induce the production of dystrophin protein in cells from this boy. And here's a Western blot. So here, this is actually a Japanese boy. He has a big deletion, 3 exons, 48, 49, 50. Still, it's the same thing. 47 splices to 51, and it's out of frame. Here's wild-type cardiomyocytes, dystrophin. Here's this boy. He makes no dystrophin. And here's a single-step direct Cas9 right here. Erase that and let skipping take place, and it brings back dystrophin. So here's a great source of our motivation. This is my neighbor in Dallas. His name's Ben, and that's his mother, and his cardiologist, my close colleague, Pradeep Mammon. There's been a lot of television shows and articles and things written about Ben and our work. One of the most poignant ones, you should read it, it's online. It's called Can CRISPR Save Ben Dupree? So we looked at Ben's mutation. Ben has a really interesting mutation. He has one single letter of his 3.2 billion nucleotides that's wrong. That's it. Put him in a wheelchair. And it's not even in an exon. He has one T in the intron between 47 and 48 exon that's converted to a G. And you think, what's the big deal? One letter in the middle of an intron. Who cares? AG. It's the universal splice acceptor sequence. So that G created a pseudo exon, a false splice acceptor. So 47th exon splices to 47A, which has a stop codon. So we can't make any dystrophin. So we looked at that, and this was another just kind of moment, where things line up. AG. Inside his disease lies his cure. His mutation is the PAM sequence for Cas9. So we made a guide RNA to direct Cas9 to just get rid of that darn G and ask if we could skip 47A and go to 48. There's the results. So these are Ben's cardiomyocytes. There's not many people in the world that get to look through a microscope and see their own heart cells beating. we had Ben come up to the lab, and this was on TV and everything, but he was looking at his own heart cells beating. There's even fewer people in the world who have a genetic disease that they're missing a protein, and then they get to see their own cells make the protein that their body can't make. So this is Ben's cells in red, they're stained for dystrophin, so now they're making dystrophin. Here's the corrected compared to his uncorrected cells. One single cut. So I'm getting to the end here. These are the types of mutations that we think can potentially be corrected by this approach. I've shown you that nonsense mutations can be corrected by skipping the mutant exon. I've shown you that out-of-frame deletions, like exon 50 deletion, can be fixed. These are partial fixes because they skip a short segment and put the protein back in frame, but the protein still works. So in this case, you're converting Duchenne, DMD, into BMD, essentially, but that would be a triumph for any patient, for sure. I haven't shown you this today, but it can also correct duplications by skipping the duplicated exon, and as I showed you with Ben, it can correct pseudo-exons by removing the false splice acceptor. And in these cases, the latter two, it's perfect. So the protein that's generated by editing of these genomes are perfect, no different from wild type. So this is where we are, what I've told you. So human cells, we can model the disease in a dish. We have made mouse models for the top 6 most common deleted exons in humans. Those mice account for about 40% of the known mutations in humans. So we're correcting each of them by AAV delivery. And the big step and the huge step, we're now trying to correct this in a large animal as a prelude to human studies. And I've been making trips here to London to do this. We're doing it at the Royal Veterinary College here of London with Richard Pierce. So, and if those things work, we would hope we could move towards eventual clinical trials, although there are many issues that will need to be addressed, and I'm happy to touch on those in the questioning if there's interest. But I think, at least in principle, and certainly in a mouse is not a man, but in principle, we think we could correct up to 80% of Duchenne mutations by consolidating them into groups that can be corrected collectively by skipping of certain exons. And so to do that, to transition this work beyond my academic lab to really make it try to happen therapeutically, we have started a biotech company called Exonics Therapeutics. So a couple weeks ago, I was looking up in the sky in Dallas, and I saw this cloud formation, which was really amazing. It's a double helix with a double-stranded break. So I twittered this out. It went viral. That's Dallas skyline with this cloud. I mean, I'm not kidding. This cloud just appeared. So I sent out spontaneous DNA cloud with double-strand break above Dallas, a good omen for CRISPR gene editing. So with that, let me just close. The work that I described today was a subset of things done in my group, the micropeptide work. I think I acknowledge the people involved. Myoediting had a great team of people that did that. In particular, Leonela Amoasi did all the viral work that I talked about. We've had great colleagues in Dallas and elsewhere, in particular Rhonda Basildubi, who helps me to run our group. This is my lab. My lab is kind of schizophrenic. I don't know. This half is the fun half. This half is the serious half. Well, he's an outlier. But anyway, on that, I'm happy to take any questions. So thank you.